Hi everyone, welcome back. In the first part, we studied about the genes responsible for POI. Next, I want to share a few thinking and perspective of POI genetics based on the latest progress in this field. First one, how to explain the pathogenicity of pleiotropic genes in isolated POI patients. Secondly, how to determine the pathogenicity of heterozygous mutations in sporadic POI. Third one is POI monogenic, digenic, or polygenic. First, how to explain the pathogenicity of pleiotropic genes in isolated POI. For example, C4, MRPS22, and BRCA2 are known pleiotropic genes responsible for some multi-system syndrome. C4 is a curative gene for Harrow syndrome, whereas its mild variants cause isolated POI phenotype. Possible explanation is that LOF mutations result in haploinsufficiency and cause syndromic phenotypes. Mild mutation identified in POI led to enhanced regulatory activity. Different mutations exhibited opposite effect. For MRPS22 gene, severe mutations will compromise mitochondrial energy production and cause syndromic phenotypes, whereas mild mutations only affect primarily in reproductive system. BRCA2 is well known for its relationship with Fanconi amenorrhea and breast tumors. Latest findings of BRCA2 in isolated POI may contribute to residual BRCA2 function. Patients with variants in pleiotropic genes might have variable presentation, which could be explained by genetic or environmental modifiers or little recognition of genotype-phenotype correlation. Genetic studies are significant in disease diagnosis and long-term follow-up. Secondly, how to determine the pathogenicity of heterozygous mutation in sporadic POI patients. By candidate gene approach, most variants were heterozygous. However, by next-generation sequence, half findings were homozygous or compound heterozygous, and the heterozygous mutation carriers in these pedigrees were normal, which questions the pathogenesis of heterozygous variants identified in sporadic POI. GDF9 and SOLR2 are two known candidates for POI, and heterozygous mutations have been reported in sporadic cases. Recently, homozygous mutations in the two genes were found in consanguineous families with POI. Similarly, the probants exhibited primary amenorrhea, and their mothers with heterozygous mutation presented with normal ovarian function. How to explain normal phenotype in POI pedigree, but pathogenic in sporadic POI for heterozygous mutations? Complex inheritance pattern, dominant or recessive inheritance. Not all heterozygous mutations are equally pathogenic. Possible interaction with other genetic defects or environmental exposure. Heterozygous mutations might have contribution to POI development, but not sufficient to be pathogenic. These are possible explanations and needs more solid evidence. Since some heterozygous mutations are not sufficient to be causative, it is proposed that POI be digenicity or polygenicity. More evidence supports digenicity or polygenicity for POI genetics. Cumulative effects of genetic defects affect the clinical severity of POI. Here are two examples. In this family, the proband with two mutations in the NR5A1 gene developed primary amenorrhea. her mother with one mutation in this gene displayed secondary amenorrhea. All three mutations impaired the transcriptional activity on target genes. Here is another family. Two doctors presented with POR. The proband carried one de novo deletion in CPEB1 gene and another missense mutation in BMP15 gene inherited from her father. This patient developed primary amenorrhea, whereas for her sister only carrying BMP15 mutation from their father. Menopause occurred at 39 years old. Findings through next-generation sequencing also provide more evidence. For example, in 100 POI cases, TGS revealed 37% of patients carried at least two genetic variants. Single and multiple mutation carriers have different age at POI onset. More mutations earlier onset. The presence of at least two mutations in distinct genes in 22% of the patients argued in favor of a polygenic nature of POI genetics. Synergistic action of variants in signaling pathway involved in ovarian function is anticipated. Those coexisting mutations in two or more POI genes in a single case could play the role of second hits or act as modifier genes involved in POR. In the last, we make a summary for today's lecture. In the era of NGS, vast numbers of variants have been emerging. New genes, unanticipated mechanism, more complex genetic architecture of POR will be revealed in the era of NGS. New strategies with NGS have been revolutionizing this field. The bottleneck is bioinformatic analysis and causative relevance validation with functional experiments or mutation-directed transgenic models. Identification of individuals with POR risk remains a challenge. Targeted panels of hotspot mutations and all validated curative genes will add important value in risk prediction, routine diagnosis, and earlier interaction. In the last, I want to express to our POR team and SDU-IVF team. Here is the picture of our group. And also I want to express my thanks to all the cooperators within China and international. Thank you all.